Hello, everyone. Good afternoon.
Hi, how are you doing?
Fine, thank you.
Good.
And you?
Good, thanks.
How are you, Dr. Croatia?
It's right, yeah.
Okay, so you are first speaker for this session, I think, yes?
That's
okay.
correct.
Okay, so, okay.
Hello, everyone. This is
Prof. Yaqub, are you moderating the session?
Yes, I co-moderated with you.
I'm quite happy for you to get us going, and then I can pick up halfway through.
Thank you very much.
Thank you.
Hello, can you hear me?
Yes, Dr. Ahmed.
Hello. This is Dr. Ahmed from University of The Hook. Sorry, Prof. Croatia and Prof. Haval. May I ask you, please, to share your cams? I'm talking about the panelists to take a screenshot for memorize, please. Prof. Croatia and Prof. Haval, your screen are already shared. Dr. Martin's screen is shared as well. Okay.
Yes. So the host is disabled. Okay, yeah. So for me, I think it's disabled.
It is okay. It is okay, doctor. I can see your camera is
yes.
on. Yeah.
Yeah.
May I ask Dr. Sharon?
Yes.
Okay, Dr. Sharon. Yes,
it's a bit distorted, right?
Yeah.
I see myself distorted here.
Okay, I can see you. I can see you. Okay. And Dr. D?
Yes, I'm here.
Okay, can you share your camera, please?
Yeah, sure. Just a minute.
Okay. All right. So, yeah. Great. I'll take this shot now.
Yes, from Marcus, maybe you can share it now. Please, could you try? Yes. Sorry for this. Can you see the slide? Yes, it's clear.
Prof. Amal, could you please share your screen with us? I'm not sure if Prof. Amal can hear me. Prof. Amal, are you with us, please? All right. Okay, let me share mine as well. All right. So I'll count to 3. 1, 2, 3. Great. Thank you so much. So, okay, Dr. Amal has joined us. Dr. Amal got to take a screenshot for the session again. One more, please. Okay.
Okay.
So all ready? Okay. I'll count to 3. 1, 2, 3.
Thanks a lot.
All right, all done. Thank you so much. You can start your session, please. Dr. Croatia and Dr. Haval, it is your time to start this session. Thank you so much.
Okay, thank you. So we're going to start, Dr. Quraisha.
Okay. So greetings, everyone. On behalf of Prof. Haval and myself, Quraisha Abdul Karim, I'd like to welcome you to this parallel technical session that will focus on health, biology, medicine, and pharmacy. We have quite an exciting lineup of speakers, and as we go along, we will introduce them. And I'm going to kick off with the first presentation.
Sorry, Dr. Quraisha. Sorry, Dr. Quraisha, but Professor Hassan Azazi will come late, a little bit late. Okay, the third speaker.
Yeah,
so that you can shift.
Thank you.
Yeah, yeah. At first, good morning, and thank you for joining us.
Yes, thank you very much. And we have Dr. Dominique on, so I think by the time our two presentations are done, we should be able to Stacey join us as well. I just wanted to check with the host if I'm able to have control of my screen.
Yes.
Okay. So I just wanted to thank the organizers for the opportunity to be able to address you this morning about COVID-19 and particularly some of the lessons that we're learning in terms of saving lives, livelihoods, and maintaining social cohesion during pandemics, disasters, and emergencies. And while I'm going to focus on HIV, I think that Iraq faces many challenges and many epidemics, including the COVID-19 pandemic. And I hope that some of the broad lessons that I will share in the next few minutes is something that can be drawn on, whether we're dealing with TB, cholera, or anything else. So in times of uncertainty, I think none of us needs to be reminded about the devastation that COVID-19 is wreaking on our communities. And just like a quick reminder, in just a matter of about over a year, we've had over 111 million cases and 5 times the number of deaths that we've seen with the Spanish flu, for example. So in responding to epidemics, in responding to pandemics and disasters, I think from this morning we've been hearing about the importance of science in our response. And part of epidemics themselves and pandemics in great detail, because while we're dealing with the global phenomena, we have a diversity of epidemics within and between countries. And with HIV, it's not been too different. For example, 70% of the burden of HIV is in sub-Saharan Africa, where 1 in 4 adolescent girls and young women account for about 25% of infections that we see can be traced back to the disproportionate burden that we see in adolescent girls and young women. Now, in South Africa, for example, where I come from, we've been able to draw on this granular knowledge and understanding with data of HIV to be able to, for example, in the Western Cape province, to be able to identify these areas where high levels of transmission of COVID-19 was taking place. And through this hotspot mapping, we're able to intervene. I think in the response to COVID-19, was not to stop the spread of the virus. That's not really possible, but was really to slow down the spread of the virus. And part of our responses in pandemics is about the centrality of scientific evidence. In the case of COVID-19, we actually have had very little information about this time a year ago. And while we've learned a lot more about COVID, about the viremic stage, about asymptomatic infection, about who develops severe disease, there's still a lot we don't know. And so as we talk about the centrality of science, it's important in COVID-19 to appreciate, in contrast to HIV, where we do know a lot to enable us to reach epidemic control. In the case of COVID-19, we've made some significant advances in terms of vaccine development. Now we have to focus on ensuring those vaccines that have been developed do not create new divisions between us, but that we ensure equitable distribution of those vaccines. We've learned for hospitalized patients the importance of oxygen. And we've also learned about the use of dexamethasone for hospitalized patients to ensure and improve survival. But as I said, to start off with, that while we try and draw on scientific evidence as much as possible, when we encounter something new like COVID-19, our knowledge continues to evolve. And even in the face of such uncertainty, we still have to respond with the best that we have. We've seen also with HIV the importance of leadership and consistent role models. Again, I've shown you some of that. But more importantly, I think when we're dealing with epidemics and pandemics is the importance of community engagement. And it's not unusual for human beings when faced with something new to respond with fear, with anxiety. But 1 year on, I think some of the stigma and fear may have been removed. I'm not sure so much about anxiety. But it's one thing for governments and states to be making decisions for its people. But when we engage our communities in responding to challenges and threats that face us, we can be a lot more effective in terms of countering the fear and anxiety. And this mantra that we've used in HIV, which is do things with people, not on people, cannot be even true, it holds true for our responses to COVID-19. And having an array of things that we can tap into is really critical. And we've seen with COVID and with HIV. With HIV, over years, it took nearly 30 years to develop a set of tools to be able to respond to the diversity of epidemics. With COVID, we've drawn on other infections with coronaviruses to start off with social distancing, isolation, use of appropriate protective equipment, and also hygiene, the use of masks, the symptom screening, and most extreme form of social distancing in terms of lockdowns. But we've also seen how technology has evolved, and some of the responses to HIV enabled us to have the infrastructure to very rapidly transition to setting up the laboratory infrastructure for RT-PCR measurements for COVID-19, and having a confirmation that somebody is infected with SARS-CoV-2 or not is an important first step, not only for individual patient care, but also in terms of containment efforts and limiting further transmission. With COVID, we've seen an unprecedented generation of new knowledge, and in less than a year, We've also had rapid point of care tests for measurements of both antibody and antigen. And while we've had some challenges in terms of reagents and securing these kits, I think at this point in time, most countries have the capacity and capability to be able to do testing. And it underscores the importance of investments that are made as we've drawn on HIV in Africa, that these preparedness activities help not only with the epidemic that faces you at a point in time or pandemic, but also creates a very strong foundation in which to transition fairly rapidly when new and emerging pandemics take hold. But I think in most pandemics that we face, there's a huge challenge, which is how do we change behaviors? And so while knowledge is important, our ability to transition and it's a prerequisite, there's a big gap between having knowledge of something with our ability to act on it. And I think we still see challenges in some countries in terms of simple things like hand sanitizer, or wearing of masks. But there are also structural challenges that face us. For example, water is so essential for containing most pandemics that face us. And in COVID-19, communities that have water insecurity, for example, something that we take for granted when you have running water, can be an immense challenge in terms of doing something basic like washing your hands and keeping your environment clean. Notwithstanding some of the structural challenges that face us, what we do know and what we've learned from other pandemics is that scare tactics don't work, and that we have to enable and empower individuals and communities to adopt the new behaviors and to transition from fears. Now, I just want to touch very quickly on gender inequities because it's a key part of our sustainable development goals. In the Millennium Development Goals and the health ones particularly, MDG 5 was about reducing maternal mortality rates, which is a very extreme form of gender inequity we see. In HIV, we have failed to bridge this gender inequity gap in Africa. For example, in Eastern and Southern Africa, young women under the age of 25 bear a disproportionate burden of infection. 1 in 4 young women under the age of 20 are infected with HIV. And in COVID-19, we started to see something very similar, where women dominate as frontline workers, but also in terms of the more resource intensive and both in the formal and informal sector, we find that there's a disproportionate burden of job losses, women having to work remotely and do both caring in the formal health environment and also at home, and the list goes on. And I hope with COVID-19, we do a better job in terms of addressing gender inequity issues. But as with HIV, we have an opportunity for universal health care. In COVID-19, data from the US and other parts of the world are showing differences in terms of racial and ethnic disparities. And what we're seeing with COVID, already vulnerable populations are bearing a disproportionate burden of COVID disease and mortality rates. But I'd like to end on a more positive note from Africa, where we've moved from anxiety to agency to looking at self-efficacy. And the key thing that drives us is the notion of Ubuntu, which is I am because you are. And this we've tapped into in terms of collective action, our interdependence, that I am safe. By me being safe, I keep you safe, and you being safe keeps me safe. So if we each do the best we can to adopt some of the COVID-19 preventive messages, we can all benefit from preventing infections from preventing loss of lives and keeping our spaces that we live and work in safe. And I just wanted to say that global solidarity is in HIV has enabled quite a large, almost 70% of people living with AIDS across the world to have access to life-saving medication. And we're starting to see the same kind of solidarity. But COVID-19, when all is said and done, is a reminder that we are one world and one people. And when we work together, we can achieve a lot. And that interdependence is what we draw on as we respond. In conclusion, we've seen both in HIV and COVID-19 how science has been transformed in various responses. In pandemic, scientific evidence has importance in decision-making, providing guidance and challenging conspiracy theories. In AIDS, we dealt with the politics of the lack of leadership and misinformation, but ultimately scientific evidence provided a path forward, example, with access to antiretrovirals. In COVID-19, we will move similarly beyond the political machinations as science develops new solutions like vaccines, and that we ensure its access. Therein lies our hope. The HIV and COVID pandemics have provided a new lens for the nexus between science, politics, and global health as we pursue the path of the Sustainable Development Goals. And I hope that as Iraq tries to rebuild its country and challenges, that we can draw on these. So let me stop there and thank you for your attention and hand over to Dr. Martin Dominique as the next speaker. He's a reader in physics and astronomy at the University of St. Andrews in Scotland, and he'll be talking about biology. Is biology universal? And what does it mean to be human? And I think it's a nice transition from my focus on COVID-19, which is really challenging us in terms of what it is to be a human. So over to you, Dr. Martin Dominique. Thank you so much for joining us, and we look forward to your presentation.
You see my slides?
We're not seeing your slides yet, Dr. Dominique. Here it comes, right. I think you're all set. Thank you.
Okay, great. Who are we? What are we doing here? Where did we come from? Where are we going? Is there anybody else out there? By studying nature, humankind explores its environment and places itself into context. The pinpoints of light on the night sky are most notable feature of nature, having inspired humankind since antiquity to wonder about its existence and how it fits into the bigger picture. There are stars that appear to be fixed to a celestial sphere, planets that move slowly, and the occasional comet or supernova. It was suggested that stars might resemble the sun, and we're supporting evidence for that since the 19th century. However, until 1995, the sun was the only star known to us to be orbited by planets. We now know about 4,000 planets orbiting other stars, and this has changed everything. And this has changed nothing. Everything about the existence of planets, but nothing about whether there's life beyond Earth or not. The sheer diversity of planets and planetary systems, which is the most remarkable finding next to their large abundance, however, yet again challenges anthropocentric theories, which have a long history of having been proven wrong. Our minds find it hard to cope not only with imagining the vastness of the universe, but along with it, also It's emptiness. Planet Earth provides an oasis that we reside in. How peculiar can it be, orbiting a star that is one of hundreds of billions in the Milky Way, which itself is one of hundreds of billions of galaxies in the universe? If the laws of nature are universal, how can it be the case that there's only one instance of life, and exactly one? Or do we simply not know better, and the universe is teeming with life all over? It was the advent of the space age with Sputnik 1 in 1957 that already opened a window to search for life beyond Earth within the solar system by means of space probes. Direct searches for life on Mars were conducted as early as 1976 with the 2 Viking landers. Now, Europa, Enceladus, or Titan have become further targets of particular interest. Realizing that radio waves allow communication even over interstellar distances, Giuseppe Cocconi and Philip Morrison suggested a search for extraterrestrial intelligence, or SETI, in 1959, while Frank Drake was already setting one up. This is highly speculative, but it isn't fiction, but reality, with the probability of success being difficult to estimate. What we know for sure, however, as Cocconi and Morrison pointed out, is that if we never search, the chance of success is 0. Many people refer to the concept of habitability as some suitability to host life, but I actually feel lost amongst the lots of definitions of habitability and the lots of definitions of life. Moreover, the links between these two concepts lack clarity. Fuzzy concepts are challenging for exact science, and ambiguous concepts are worse. We're close to the point at which nobody really understands what the other one is talking about. What do we actually? In 2014, the European Research Council hosted a session at the Euroscience Open Forum, ESOF, for which it issued a press release stating that Earth is the only habitable body in the solar system. This got me confused, given that this is either trivial. If one defines habitable as having properties that only natural, or it is wrong.
I'm sorry. Can we please have everybody muted except the speaker? Thank you.
So
Go
it's either
ahead.
trivial if one defines habitable as having properties that only Earth matches, or it is wrong if habitability is considered a requirement for life to exist. Feel free to take your pick. Moreover, there's no evidence that permits to rule out life on any planet. So all of them are habitable? I think that such communication issues could be indicative of underlying problems with concepts or adequately framing research questions. Habitability can be seen as a proxy for the probability that life exists, where the latter is no more than a reflection of our Earth-centric prejudice, which sets prior assumptions. It thereby is a measure only of our expectations, while reality may be entirely different. A rather patchy and fuzzy knowledge is not well mapped to draw a system which requires to draw a sharp line between planets that we might want to call habitable and those that we decide to call uninhabitable. It would seem to be that we actually made very little progress over the last 350 years on getting concrete. John Wilkins, one of the founding members of the Royal Society, speculated about life on the moon in the 17th century. notably using the term habitable with all its surrounding fuzziness in a most British fashion. His treatise, Discovery of a New World, is labeled as a discourse tending to prove that it's probable there may be another habitable world in the moon, which appears to be more honestly phrased than the many sensationalist statements that were made over the last decades. While habitability has been seen as a means for guiding exploration, we now know that planets exist pretty much wherever we look, and planetary systems show great diversity. This challenges both the usefulness and the need for the concept of habitability. So let's talk about life. I think that a key reason why attempts towards a unified definition of life have failed is that at least two very different concepts are being conflated and should better be separated. We can look at the collective process and ask, is there life? And also at individual beings and ask, is this a life? This can lead to contradictory answers. So I would want to argue that we should think about abandoning the concept of a unified definition of life and work with two different concepts, here labeled e-life and I-life, for evolutionary and individual respectively. Consider in particular the hypothetical case of individuals living forever, which would kill evolution. That is, eternal I-life would come with no e-life at all. Maybe we need even more than just two concepts, and it might be sensible to adopt definitions that are in conflict with some of our historic understanding of life. While the laws of thermodynamics govern physical and chemical processes, and in particular the entropy increases, we suddenly see life coming up and evolving a la Darwin, while with entropy locally decreasing. This is not forbidden, but why should it happen? And under what conditions should it happen? Tracing back our evolutionary history to the origin of life still leaves us with a singularity without understanding why it occurred. There are two potential pathways, however, to shed light on the emergence of life: detecting many instances of life and reading off the conditions, or finding the fundamental principle of nature that drives the transition, that is, finding a framework that unifies thermodynamics with Darwinian evolution and explains why and under what circumstances life emerges. Claiming the existence of life simply as being the only intelligible explanation for observations within current knowledge would be a fallacy that has historical precedence. In 1877, Giovanni Schiaparelli observed and mapped an elaborate system of channels on Mars, which was strongly promoted as case for intelligent life by Percival Lowell. It took about 25 years to be dismissed as a result of an optical illusion, which had hitherto been beyond current knowledge. The ability to obtain fingerprints of molecular abundance in the atmospheres of extrasolar planets is most exciting from a geological perspective. What this could mean for such signatures hinting at life on such planets is actually not clear at all. Let's, for example, look at the Earth's oxygen history. Strikingly, life sustained for about 2 billion years with very low amounts of oxygen. Then oxygen rose to a higher level and stayed there for another billion years. And only for the most recent 600 million years, there was as much oxygen as we find today. This history is an interplay of evolving life with the very specific structure of planet Earth, the oceans and rocks affecting the atmosphere. Oxygen produced by cyanobacteria could be bowed until the natural oxygen got saturated. During this initial period, evolution was rather slow, and the catastrophic event of a sudden rise of oxygen levels came with a mass extinction of monocellular organisms and the rise of multicellular organisms. This shows that the emergence of multicellular and higher organisms was not a straightforward consequence of life emerging on a planet. There are further hurdles with their own specific conditions attached. Moreover, while we see evolutionary convergence, the mass extinction of species through catastrophic events reshuffles the cards and breaks the chain of predictability within a controlled environment. We should therefore not jump from astronomy to biology without taking into account geology. We also see that monocellular life can happily exist for a long time without substantial amounts of oxygen arising in the atmosphere. And the animals were the evolutionary winners with the later rise in oxygen. So life can easily escape detection. Very simple setup demonstrates nicely how challenging any detection of life by remote sensing is. We know that there's life in a pot of yogurt, but imagine you put it at just 1 meter distance from you. You're not allowed to touch it or get a probe. Your only means is electromagnetic radiation. This life so close. How do you prove that it is there? Finding life is hard. Not finding anything could still mean that we're far from being alone in the universe. If there's only one life-bearing planet in the Milky Way, and this is typical across the universe, we're in a cosmic company of hundreds of billions of others, but wouldn't know. Exploring the universe in the search for ourselves is a venture that is most fascinating and most frustrating at the same time. What does it mean to be human? A question that cuts across all three classical pillars of philosophy, natural philosophy, metaphysics, and ethics, was central to an exhibit, A Message from Afar, showcasing the search for extraterrestrial intelligence at the 2019 Royal Society Summer Science Exhibition, which I designed and led for the UK SETI Research Network. It is in fact the deeper question behind the popular Are We Alone?, which is the question of natural philosophy alone. What distinguishes a signal from a message? How natural is intelligence? Is communicating information a fundamental principle of biology? Visitors could explore by means of colored cubes how to transmit a message by designing their own code, and they were given a range of examples of how we encode different language and terminology, which constitutes a substantial obstacle to progress. Withstanding pressure to provide answers, we moreover require clarity about what we don't know. There's a lot of wishful thinking around, while many lines lack connections. Many research agendas that I've seen on this topic remind me of the famous cartoon by Sidney Harris, and I would never buy a roadmap that only shows one road. An upcoming project of the network of researchers on the chemical evolution of life, short NORCEL, is to produce a gap map, which, in contrast to a review that discusses what we know, aims at clearly spelling out what we don't know about the universality of biology. Brainstorming on this will start as part of our upcoming network meeting on the 29th and 30th of March, for which registration is still open and further participants are much welcome. NORCEL thrives from the diversity across its members in all aspects of the world and aims to be a truly global endeavor and not one that just connects Western Europe with North America, Japan, and Australia. Bringing together creative minds who dare to think beyond well-trodden paths and are willing to engage in respectful discourse in order to explore potential new directions and build collective endeavors. We particularly welcome and value engagement by students, respecting only the power of the better argument, not the power of authority. Let's do science that excites, inspires, and delivers. Thank you.
Thank you very much, Dr. Dominique. Really quite the tour de force about life and being human. And thanks also for making the announcement for your upcoming meeting. So we're just going to move on with the next presentation, and we move now from the world of physics to and astrophysics to biology again. And we have Dr. Shen En Un from the University of Malaysia, who will address us on molecular targeted therapy, honing in on cancer with precision. So I think Dr. Amal is not with us, but only she will put together a exciting and varied and diverse session as this. So Dr. Un, we look forward to listening to more targeted approaches to cancer therapy. Thank you.
Thank you very much. Can you hear me?
Yep.
Okay, great.
And we can see your slides as well. Thank you.
Oh, great. That's great. So good evening from Malaysia. I'm Chern. I'll be talking to you on molecular targeted therapy. When you think of molecular targeted therapy, what do you think? Can you just imagine this scenario in what used to be a peaceful village? Suddenly, there was an invasion of intruders, aka the cancer cells. It may have started with one trespasser in disguise to blend in with the rest of the villagers, and slowly the uninvited intruder multiplied and brought in the rest of its kind in the bid to attack and take over the village. The security forces got called in. Targeted therapies are likened to the security forces. Their aim is to selectively kill the intruders without causing much harm to the civilians or the healthy cells, but only to gun down the intruders. So this is how the molecular targeted therapy works. Precision medicine is the future of healthcare. It targets different patients based on their unique circumstances by taking into account the genetic profile and environment and lifestyle factors. The genetic variations in each individual may affect how a patient responds to a certain drug. Environmental factors and individual lifestyle have also been shown to influence gene expression and how an individual responds to treatments. The patient's genetic information are then used to predict an individual's response to certain drugs by taking into account the environmental factors and to assess the risk of developing particular diseases. So in short, precision medicine allows patients to be treated by tailoring therapies according to their genetic makeup, and this allows the patients to receive the right treatment at the right time. Molecular targeted therapy is the cornerstone of precision medicine. So molecular targeted therapy can come in different forms. It can come in the form of small molecule inhibitors, monoclonal antibodies, cancer vaccines, or gene therapy. Basically, molecular targeted therapy works by targeting very specific molecules that are highly expressed in the cancer cells, but not in the normal healthy cells, thereby only destroying the cancer cells without harming the healthy tissues. In drug discovery, once a molecular target has been identified, in our case, the sirtuin enzymes, the next step is to develop a therapy that affects the target in a way that interferes with its ability to promote cancer cell growth or the survival. A targeted therapy could reduce the activity of the protein target or prevent it from binding to a receptor, or it can come in the form of monoclonal antibodies. Small molecule compounds are typically developed for targets that are located inside the cell because such agents are able to enter the cells easily. However, the antibodies are relatively large and generally cannot enter the cells, so they are used for targets that are located on the surface. So altogether, they work by inhibiting the downstream events to inhibit cell proliferation, angiogenesis or the formation of blood vessels, cell invasion and metastasis, cell cycle regulation, or cell survival. The tumor microenvironment can potentially reprogram tumor initiation, tumor progression, invasion, metastasis, and responsive therapies. Therefore, research focus on cancer treatment has switched from a cancer-centric model to a tumor microenvironment-centric one. So what are sirtuins? Sirtuins are highly implicated in metabolic diseases, including cancer, diabetes, neurodegeneration, and cardiovascular diseases. Sirtuins are class 3 histone deacetylases. They need nicotine amide adenine dinucleotide or NAD+ as cofactor. They basically have deacetylase and or ribosyl transferase activities. They are located in different subcellular compartments within the cells, and this leads to the diverse biological functions. Sirtuins are highly conserved from yeast to mammals. In mammals, there are 7 sirtuin homologs, SIRT1 to SIRT7, and SIRT1 is by far the largest. Therefore, it controls the most downstream activities. They share a conserved 275 amino acid core, and they're being flanked by the N and the C terminals of variable lengths. And these variable lengths have, and these N and C terminals have been shown to affect the activity of the sirtuin enzymes. Sirtuin inhibitors to treat cancer remains one of the most hyped areas of present-day research. Okay, several classes of sirtuin inhibitors with a broad range of core structures have been identified, and they include X527, which is a very specific SIRT1 inhibitor, AGK2, a very specific SIRT2 inhibitor, nicotinamide, sirtenol, tanovinsalamide, suramine, and cambinol are all SIRT1 and SIRT2 inhibitors. However, these sirtuin inhibitors vary in stability, potency, and target selectivity, mainly due to the chemical modifications as well as the complex role of each sirtuin member. Further development of these modulators are so far unable to reach clinical trials for cancer treatment due to either unfavorable toxicity in vitro, poor physiochemical properties, or unexpected promotion of tumor growth in vivo. So far, X527 is in a phase 3 clinical trials, but for the treatment of Huntington's disease. Sirtuin is complicated. It can function as a tumor suppressor or a tumor promoter, and this is very much dependent on the mutation profile, the tumor grade, or even the location of the sirtuin or the tumor microenvironment. We are interested in sirtuin-1 and sirtuin-2 because they are highly expressed in acute myeloid leukemia, colon cancer, prostate cancer, pancreatic cancer, stomach cancer, and melanoma. We have patented a small molecule inhibitor, which we name as BZD9L1, and this inhibitor can inhibit SIRT1 and SIRT2 activity to induce apoptosis cell death in colon cancer. This discovery was first made in 2013. The compound itself is autofluorescent and it has a very high selectivity towards colon cancer cells compared to normal epithelial cells. And at this stage, our compound is in the preclinical studies. As I've mentioned previously, the compound is autofluorescent. So upon treatment with the compound, it diffuses into the cells. And this is an added advantage for this drug to be used in pharmacokinetic or in vivo imaging studies. So how does BZD9L1 work? It works by competing with the coenzyme NAD+ in SIRT2. So basically, it competes with the binding region of NAD+ and displaces NAD+, and therefore this inhibits SIRT2 activity. In vitro, when tested on a panel of colon cancer cell lines with different mutation profiles, together with the other commercial inhibitors, BZD9L1 was found to fare almost as well as tenovin-6. So tenovin-6 is our closest inhibitor, competitor. However, BZD9L1 showed a higher selectivity index compared to tenovin-6, meaning to say that our inhibitor is less toxic to the normal colon epithelial cells when compared to the colon cancer cells. Very interesting observation has been made. The compound was able to modulate cancer pathways differently in both HCT116 and HT29 colon cancer cells. So these two cancer cell lines are colon cancer cell lines, but with different mutation profiles. However, they ultimately cause the cancer cells to undergo apoptosis cell death. When combined with 5-fluorouracil, which is the cornerstone treatment, chemo treatment for colon cancer, it synergistically reduced the HCT116 cell viability, but this was not seen in the other colon cancer cell line, HT29. The combination treatment also resulted in a reduced spheroid migration, but not invasion. In the animal model, the combination treatment reduced the xenograft tumor growth compared to sole treatments Very interestingly, the body weight of the animals in each of these treatment groups remained unchanged. The reduction of the xenograft tumor growth was found to be through the reduction of tumor necrosis as well as tumor proliferation. So in conclusion, BZB9L1 was able to inhibit SIRT1 and SIRT2 activity to modulate the cancer pathways differently, but ultimately induce apoptosis to kill the colon cancer cells. And in combination with 5-fluorouracil, it was able to induce apoptosis and inhibit spheroid viability and migration. And in the animals, it was able to reduce tumor growth. Ongoing work involves further studies of this compound and see how it modulates the blood vessels as well as the cancer-associated fibroblasts. We are also in the midst of studying its toxicity profile in vivo and its metabolic toxicity in the liver as well as kidney. The take-home message is that sirtuin's function varies in cancer and it may very well be tumor or mutation specific. Therefore, its role is to be examined on the basis of cancer mutation profile and the microenvironment. So I believe that the future is already here. Effective cancer therapy may not be dependent solely on tumor type, but based on tumor mutation. A recent study in Lancet Oncology has revealed that VS6766, which is a drug in phase 3 clinical trial, or is it phase 2, sorry, can inhibit signaling and proliferation of different tumor cell lines with a variety of very challenging cancer mutations that include KRAS, HRAS, and BRAF mutations. And in many cancer types with these mutations, it has been shown that they have very limited treatment options. So with this, I end my presentation. Thank you for your time.
Thank you very much, Dr. Un, for that really inspiring presentation. And it ends on such an optimistic note. I think cancers have been dominating our health landscape in a kind of way that was almost inevitably fatal, and to start to see so smart interventions coming in. It also reminded me to wish all the women panelists, as well as attending the session and this meeting, a very happy International Women's Day. And I think you're starting to see when women are given equal opportunities to contribute in all facets of life, how much value add and synergy is possible. I also want to extend a very warm welcome to Prof. Hassan Azizi. And with that, I'll hand over to my co-moderator, Prof. Haval, to introduce the next two speakers. Thank you very much, Prof. Haval.
Thank you, Professor Qureisha. I just would like to remind you that every speaker has 15 minutes, and the discussion with the question will be at the end of the session. So our next presenter is Professor Hassan Azazi from Egypt, who will present LMOF optical chemosensor for instance detection and trapping of mercury ions in water. So please, Dr. Azazi, the floor is yours.
First of all, thank you very much, Professor Haval, for your nice introduction. Also like to thank Professor Amr Amin for her kind invitation for me to join this interesting meeting. And of course, congratulations to all ladies in this conference and globally for the World Women's Day. Today, I'll talk to you about the aluminum MOF, or aluminum metal organic framework optical chemosensor for instant detection and trapping of mercury ions in water. My name is Hassan Azazi. I'm a professor and chairman of chemistry department at American University in Cairo. This presentation will include a background and then the details of the development of the TAM chemosensor, and then an attempt to move these mercury chemosensors from lab to market, and then finally a summary. The inspiration of this work is clean water is a human right. And according to the United States Environmental Protection Agency, water may be contaminated either by physical or chemical or biological and radiological entities. And some of the chemicals include heavy metals. The following heavy metals like arsenic, cadmium, lead, and mercury may contaminate water. And today we'll focus our discussion on mercury. Mercury actually is among the World Health Organization top 10 chemicals of major public health concern, and in this include very dangerous chemicals, and mercury is one of them. And of course, there are 3 other heavy metals, arsenic, cadmium, and lead, also among this list. We can be exposed to mercury intoxication either by breathing contaminated air, or drinking contaminated water with mercury, or eating fish, or from the amalgam fillings in teeth, and some vaccines which may include mercury as an additive or a preservative. And of course, some cosmetics, especially the skin whitening cream, may contain high concentration actually of mercury. Mercury ions will be readily absorbed and react with the sulfur groups of proteins, causing a change in their tertiary structure and loss of activity. Mercury ions also may get concentrated in the kidney during clearance and causing nephrotoxicity. Methyl mercury is highly selective for lipid-rich tissues like neurons and is very toxic. Water contamination with mercury may result from anthropogenic release, like from mining operation, petroleum refineries, electronic manufacturer, and burning fossil fuels, especially coal. Also, via the waste disposal sites. It could be geological due to natural mineral deposits rich in mercury. Contamination of private wells may happen through groundwater movement as well as surface water runoff. Continuous exposure to mercury will result in their bioaccumulation in the body and extensive damage to multiple organs and systems. Toxic metal detecting use the reference method for this are usually something like atomic absorption spectroscopy or ICP-MS. These usually are central governmental labs, but these usually are complex and with high cost, sophisticated infrastructure, and they need highly skilled technician and long turnaround time. We have developed thioketone derivative aluminum metal organic frame chemosensor for detection of mercury ions. We call them the TAM chemosensors. These can be prepared by reacting aluminum nitrate with terephthalic acid. This will result in the formation of the aluminum MOF nanorods, and this can be loaded with probes for detection of mercury. In this case, this will be a thioketone derivative. Interaction of the thioketone derivative with mercury ions in water under optimal condition. This will result in a change in the color from faint yellow to dark green, depends on the concentration of mercury ions in water. So this is the structure of terephthalic acid and aluminum nitrate, and this is the probe, which is a thioketone derivative, which will be responsible for capturing and interacting with the mercury ions in water. This is a proposed mechanism for the formation of nanorods or the TAM chemosensor. First, we have the reaction with aluminum nitrate and terephthalic acid forming aluminum MOF nuclei. Within 6 hours, we have horizontal growth. And then after 12 hours, we have the vertical growth of the nanorods and the formation of the TAM nanorod chemosensor. These are images of the TAM chemosensors using the FE-SEM as well as TEM. You can see the nanorods has a length of around 300 nanometers. We also characterize the TAM chemosensor with regard to nitrogen absorption and the pore size, RD, wide angle and small angle, and compare sensors before and after mercury ion in water. We also have evaluated the optimal condition for the binding of the TAM chemosensor to mercury ion in water, and this is with regard to the amount of the sensor. We found that the 10 milligram and sometimes 20 milligram of the sensor will be optimal for binding of mercury ions, regardless of the concentration. And then a pH of 7 is also optimal for binding. We have done also interference study to test the ability of the TAM chemosensor to specifically detect mercury in presence of other heavy metal ions like cadmium, like copper, and lead, and zinc, and so on. And we can find very high selectivity for the detection of mercury ion in water. We have also analyzed the absorption spectra of the TEM chemosensor at absorbance at 610 nanometer, as again under optimal condition of pH 7, measurement for 1 minute and 20 milliliter of volume of water and 25 degrees Celsius. We also have established a calibration curve for the TEM chemosensor, and we can find a linear range between 2 times 10 to the power negative 9 to 2.1 times 10 to the power negative 6 molar, and the lower limit of detection is around 0.8 part per billion, which is actually lower than the limit or the cutoff limit allowed by the WHO. These are the parameters for the mercury TEM chemosensor, pH 7, lambda max at 610 nanometer, amount of sensor around 10 milligram, response time within 30 seconds, which is very fast. We have instant response to the presence of mercury in water by the change of the color from yellow to green. And again, the detection limit is 0.8 part per billion, which is lower than what's allowed by the WHO, which is 2 part per billion. This is a comparison of the linear range as well as the detection limit of the TEM chemosensor developed in this study and other chemosensor developed by other groups. And you can see it compares very well with regard to response time, linear range, as well as the detection limit. By the way, all of this work has been published in the SES Sustainable Chemistry and Engineering paper, and I'll be happy to send this paper to any of you who are interested to know more details about this work. Again, we have tested, also evaluated the ability of the TEM chemosensor for their ability to absorb large quantities of mercury, and actually the adsorbent capacity of this chemosensor actually it's around 1,110 milligram per gram, which is actually much higher than any other removal capacity reported by other material that have been used as adsorbent for removal, for capturing and removal of mercury ion from water. We actually have modified the protocol for detection of mercury ion, not in water, but in cosmetics, because some cosmetics actually commercially available contain high concentration of mercury ion, especially the skin whitening cream. And in this case, we treat that cream with a nitric acid followed by filtration. And then we add around 20 milligram of the TEM chemosensor and adjusted the pH to 7. And then if within 30 seconds, we can change in color from yellow to green, indicating the presence of mercury ion in these cosmetics. We have tried an attempt to mobilize this work from lab to market, and we have developed another form of the mercury chemosensor by loading filter paper with nanosilica, then added the thioketone derivative, which is responsible for capturing and detection of mercury ion, and then the presence of mercury ion in water or in other media will result in change in color. As you can see, we have developed a mercury sensor. You can see a change in color from these strips, which are now try to mobilize out of lab from yellow to green. We also have developed another sensor for cadmium, which is change color from yellow to pink. And also we have developed a third one for detection of lead, which is change color from yellow to dark brown. These strips are capable of generating results instantly. They are selective and sensitive and of low cost and easy to use, and can be deployed as a part of a portable system for on-site detection of these heavy metals in water. We have also developed a digital colorimeter, which can quantify the level of the mercury or other heavy metal in water. So essentially, we read these strips with the water and then detect whether or not we have a heavy metal. Then if we start seeing color, we can use this digital colorimeter to quantify the level of heavy metal in water. So this, in a way, this is the Toxicity Skid. This is what we call it. It has the chemosensor strips. We have a specific chemosensor strips for each of the heavy metal, cadmium, mercury, and lead. Then it has a plastic cylinder to take a water sample. There's a filter to clean the water, testing container, buffer solution in order to adjust the pH to 7, which is optimal for the TEM chemosensor to detect the mercury. We have also a color chart to indicate the level of the color, and it can have the handheld digital colorimeter to quantify the result generated. We have a patent that has been granted by the United States Patent and Trade Office back in 2019 for the innovative development of these chemosensors, which are highly selective and sensitive. And we have several publications. All of this work that I presented you today is based on our publication in SS Sustainable Chemistry Engineering, which was published last year. And in summary, we have developed a low-cost optimal chemosensor, which is composed of the thioketone derivative aluminum metal organic framework for sensitive and detection of mercury ion in water. The optical signal generated is instant within 30 seconds. We can see change in color from yellow to green. The sensitivity of the developed sensor is 0.8 part per billion, which is lower than the World Health Organization permissible limit. Superior absorption capacity of 1,110 milligram per gram, and this sensor is stable and can be used several times with high efficiency. The TAMC optical chemosensor has great potential for detection and trapping of mercury ions and can be assembled in a portable kit for on-site detection of heavy metal in wells and other sources of groundwater. Definitely the reduction of mercury and exposure to mercury in the environment support the sustainable development goals of the United Nations, and especially these listed 8 out of the 17 SDGs of the United Nations. Finally, I would like to acknowledge the American University in Cairo for funding this project. And of course, the real heroes in this work, which are my research team, Dr. Ahmed Abbadi, Dr. Islam Al-Suwaifi, Mr. Ahmed Radwan, and Engineer Ali Magdi. Thank you very much for your kind attention.
Thank you very much, Professor Azazi. I hope you all enjoyed this amazing presentation. Now we move to the final presenter. We will hearing a presentation from Dr. Ritu from India. Will be speaking to us on a metagenomic analysis of gut resistance of diarrheal patient in Kolkata. Reveals diarrheal gut as a reservoir of antimicrobial resistance. So the floor is yours, please.
Hello, everybody, and a very good evening. It's evening in Kolkata, India, from where I'm talking. And the topic for my presentation is, you can see it's metagenomic analysis of the gut resistome, which we conducted using fecal samples from diarrheal patients admitted to the infectious diseases hospital in Kolkata and also at the outpatient ward. But before I go into the study, I would first like to give you a brief background why we undertook this study. So we know that antimicrobial resistance is one of the major problems in the healthcare regime today. It is challenging the efficacy of antibiotics which are required for the treatment of infectious diseases. It is leading to failure of treatment and rise in the number of deaths due to antimicrobial resistant pathogens. And one of the major reasons is antibiotic consumption has increased by 36% in the in the 21st century, and this problem is not just restricted to any particular country, but it is a global challenge. And one of the reasons why AMR problem has really emerged is because of the overuse and misuse of antibiotics. So many times, especially in countries like India, we see that we can just go and fetch antibiotics over the counter without even the prescription of professional medical, without the prescription of a doctor. And a person, even if it's not required, they have been found to take antibiotics, and this has given rise to this problem. And antimicrobial resistance, as we know, arises when the organisms, they acquire resistance determinants, which help it to develop resistance against these antibiotics, and they become extremely difficult to treat or to kill, and the infection gets extremely difficult to cure. So, and these ARGs, as we call these antimicrobial resistance genes, that is encoding resistance against these antibiotics. So they are found in different ecosystems, in different reservoirs in the environment, and also it is found in the gut of animals and humans. And antimicrobial resistance can be acquired by the organisms from one another, across species, across genera, across families, and even across different phyla. And and they are acquired by different mechanisms. They are another way that antimicrobial resistance arises is when they are inherited from parent to offspring by vertical gene transfer. But there are very few resistance genes which are chromosomally acquired from parent to offspring. But majority of the genes are actually horizontal. They are transferred by horizontal gene transfer from one organism to another, and that is the way that broadly the genes get spread and distributed in any ecosystem. So here is a rough global estimate that we predict that AMR will take a huge toll. By 2050, it is estimated that especially countries of Asia and Africa will be the worst hit, worst affected. Right now, the CDC counts the stand at 2.8 million cases in America alone, in the US alone. That 2.8 million cases of AMR infections and 35,000 deaths annually. But by 2050, it will go up, and it will take a real hit a very bad picture globally. And like AMR, it is going to affect so horribly that infectious diseases like diarrhea, tuberculosis, pneumonia. Tuberculosis, pneumonia has already been badly affected, and the diarrheal diseases, which is one of the leading causes of death worldwide, especially in the middle and low income countries, and it is the second leading cause of childhood mortality around the world. It accounts for 5,025,000 deaths per year. There are 1.7 billion cases of diarrhea every year, and the major diarrheal pathogens like Campylobacter, Escherichia coli, they have been found to be like almost 100% drug resistant. According to CDC, these are some of the biggest, the urgent threats like Campylobacter, serious threats like Escherichia coli. And in India, the majority of the diarrheal diseases are caused by these organisms like Campylobacter, Escherichia coli, Vibrio cholerae. And there are like 3,34,000 childhood deaths per year in India because of diarrheal diseases. So for us, for countries like India, antibiotics, they hold a major place, because without antibiotics, we cannot treat the population suffering from diarrhea. But at the same time, AMR is a major problem with the diarrheal diseases, and diarrheal diseases will become untreatable. There will be more number of deaths due to AMR. So to look into like from where the AMR genes are coming, the ARGs are coming, what are the reservoirs, in order to like with the aim of finally finding an intervention target or intervention strategy for alleviating the problem of diarrhea and AMR together, we undertook this project. So I'll give you a brief overview, like what kind of drug resistance pattern we are finding in two of the major diarrheal pathogens, Vibrio cholerae and Shigellosis. Cholera is a major problem in India. There are a lot of cases of Vibrio cholerae that are isolated, and here is a panel of antibiotics which we are recommended for the treatment of cholera. And here I have the data from 2015 to 2019, where you can see that like Vibrio cholerae isolates, although the problem is not as pressing as in E. coli or Klebsiella or Campylobacter, but still we have a big problem when it comes to nalidixic acid, then streptomycin and SXT, sulfamethoxazole trimethoprim. And Shigellosis, we know like almost, I think if I remember the correct figure, 69% of diarrheal cases are due to Shigella in the world. And in India also, Shigellosis is a huge problem, especially because for the childhood pediatric population, it is a major cause of childhood bacillary dysentery. And here Shigella is like The picture is really horrible, as you can see. And from 2015 to 2019, we can see that majority of the antibiotics, Shigella, the percentage of isolates that are resistant to these antibiotics, it's like almost 100%. Especially like ciprofloxacin, broad spectrum antibiotics, Shigella, we found that almost 100% isolates are resistant to these antibiotics. Therefore, our objective, as I already told you, it was to identify the AMR determinants in the gut microbiome of diarrheal patients, and to also identify the source meaning the organisms from which these AMR determinants might be coming from. So we undertook two strategies. One was based on simple molecular typing techniques. We conducted a PCR Sanger sequencing based targeted approach, where we selected a panel of 48 major ARGs, like antimicrobial resistance genes, which are resistant, covering a panel of eight major classes of antimicrobials, and they're involved in different mechanisms of AMR. And we selected these ARGs, like the common ARGs that are usually encountered within the diarrheal pathogens. And then we did a PCR screening to determine which ARGs are present and which are absent. Once we identified or detected an ARG, we did a genetic identification confirmation based on Sanger sequencing. And then from those 25 samples, diarrheal samples that we screened for the presence of these 48 ARGs, we selected a subset of 10 samples where we conducted another PCR assay to determine the antimicrobial resistance determinants against the beta-lactam and carbapenems. And later, once we detected these antibiotic resistance genes, we wanted to study the structure of the microbiota to get an idea that which might be the source of these genes. So we undertook NGS technology, next generation sequencing technology, using metagenomics to study the gut microbiome of these diarrheal patients. And later we used whole genome shotgun sequencing to determine the resistome. It was a targeted, like we selected the genes when we conducted PCR and Sanger sequencing, and resistome would give us the full complete overview of what all resistance determinants, or at least the classes of antimicrobial resistance determinants which are present, the determinants against which classes of antimicrobial resistance are present, a full view. So for the first approach, the PCR and Sanger sequencing based approach, here is like, these are the classes of antibiotics, and these are the ARGs against each of these classes of antibiotics which we tested. And here are representative gel pictures. We also had included MGs, that is mobile genetic elements, because many of these ARGs are born on the mobile genetic elements like plasmids, transposons, and integrons, and that is why they are easily transferred from one bacteria to another. And so here like I'm not showing you picture of the for all the, here, just a few. As you can see here, it is integron 1, marker for integron 1, for integron 2, and integron 3 here, integron 4 here. These are usually encountered in clinical strains, these integrons, and we found them in the fecal samples of diarrheal patients. And here we found a number of tetracycline resistance markers. So here is the data, like all these 25 patients. Here they were admitted to the hospital, and here is the profile that of the resistance genes that are present on the basis of PCR that we detected. And here are the pathogens that were isolated by classical microbiological method associated with each diarrheal sample, and which are responsible for causing diarrhea in that particular patient. And we have a number of mixed infections also. But we were not very convinced that all these antimicrobial resistance genes are coming only from these pathogens. So we wanted to undertake a bigger study and type, or rather study the entire microbiota and the entire resistome. So based on the PCR, we found that we calculated the abundance of individual genes that are circulating in this population. And we found that the major problem like MEF, AIM, MPHA, these are encoding for resistance against macrolides, and they are present in the gut of 96% patients, and then followed by tetracycline resistance. These are the markers for tetracycline resistance, and they are found up to 84%. And here are the markers for the macrolide resistance in this blue. And and here we have STRI-STRAB, which are encoding resistance against streptomycin, and they are active against aminoglycosides, and these are all very broad spectrum antibiotics and very, very important for the treatment of diarrhea. Later, we assessed or estimated the frequency of occurrence of resistance markers against each antibiotic class, and we were really shocked when we found that macrolides had the resistance markers against macrolides had occurred in the gut of 100% patients, and these are very shocking figures and really like ominous figures, which really help us to predict that we are heading towards a horrible crisis in the treatment of diarrhea. Isolated, we were able to detect TEM, OXA1-like, and DHA genes, which are encoding resistance against against the broad spectrum antibiotics, beta-lactam group. Here we found that OXA1-like was of highest occurrence. 30% patients, they were in the gut of 30% patients. We found the presence of OXA1-like. In 20% patients, DHA and TM. They are all encoding resistance against beta-lactams and carbapenems. Later, after this, we moved on to metagenomic sequencing. We took 20 samples, and each one associated with its own diarrheal pathogen, which was responsible for causing diarrhea in these patients, and we went in for amplicon sequencing. And we found that the major phyla that was present or in the diarrheal gut, they were like mainly actinobacteria. Here we have the firmicutes, the actinobacteria here in green, firmicutes, and here the bacteroidetes. And we found that in diarrheal patients, firmicutes is usually higher in abundance than the bacteroidetes. And then we have a huge proportion of proteobacteria. And then we here I have not presented the class abundance, but family. At the family level, we found that major families contributing or present in the diarrheal gut were mainly Streptococcus in pink, Enterobacteriaceae, a huge proportion here in red. And here we have bifidobacteria. Although bifidobacteria, they are mainly commensals and are used as probiotics, but this is like the structural composition. And then we tried to plot this beta diversity, studied beta diversity to see if based on the diarrheal pathogen associated with each fecal sample, if we can group or if they are showing any particular assembly or grouping. But we didn't find anything as such. They were all spread out. Based on like no particular, no fecal sample associated with any particular diarrheal pathogen was found to have the same kind of structural composition. And here is the heat map where we found the proportion of each of these families of bacterial families in each of these fecal samples. Here we find that in many of these samples, as I already presented to you, Streptococcus was very high. Then we moved on to whole genome shotgun sequencing to study map the entire resistome. And we found, like it was quite matching with the data of our targeted approach, that tetracycline resistance was quite high in all these, in majority of the samples. And then beta-lactam and quinolones, beta-lactam in green and resistance determinants against quinolones and macrolides, macrolide in this color. And we also found the biosynthetic gene clusters, and we found that bacteriocin occurrence was of the highest abundance, followed by the other non-ribosomal peptides. And then we wanted to, with the help of from these assembled genomes, we could identify that which organisms are majorly contributing to this diarrheal gut resistome. And as we can see here actinobacteria. And we would be quite shocked to see that it's bifidobacterium. The commensals have also become quite drug resistant. I mean, even we cannot say if they are drug resistant, but they are harboring the genes, antimicrobial resistance genes against these major antibiotics. The phylum Actinobacteria represented in green, and here are the different species of bifidobacterium. And also, like a huge proportion is coming from proteobacteria, and here we have Klebsiella pneumoniae, Escherichia coli, which are very important residents of the commensal gut microflora. And if these are resistant, so the next fear is they are going to transfer these genes to even other resident microbes and also the invading pathogens. And so diarrheal diseases will be extremely difficult to treat. So in summary, I would say that we found high abundance of ARGs against the major classes of antibiotics, including the beta-lactams, the carbapenems, and the sources of ARGs could be traced to families like Enterobacteriaceae and Streptococci, and in major residents of the gut microflora like Klebsiella and E. coli, and which are with E. coli and K. pneumonia, like belonging to family Enterobacteriaceae, there are exchange communities. Like with Vibrionaceae, they exchange a lot of these genes, and also they exchange genes with other species under the same family, like Shigella, Salmonella. So as it is, these bacteria have become multi-drug resistant, and in the future they are going to cause more grave serious problems. And so how does it contribute to public health? We were able to identify, this was a small pilot study that we undertook, and it helped us to identify the sources of AMR in the gut. And therefore, in the future, we hope that it will help us to determine or design some intervention strategy to stop the dissemination of AMR. I would like to acknowledge a few people. The study was supported by the Department of Health Research, and thank you.
Thank you very much for this informative and interesting talk. We would like to thank all of our speakers for addressing this interesting topic. Now, if anyone has a question, I'm sure all speakers would be pleased to answer them. And you have two options to ask your question. Either use the small icon, raise hand, or you can write your question on a chat or question and answer. But please state to which presenter you address your question. I can see one question on the question and answer port. Okay, I think all of them has been or have been answered by the speakers. So.
It seems to me, Prof. Yaqub, that I think our presentations were so clear. Everyone
yes,
knows
we have. Sorry, Professor Qureisha, we have one hand raised, please. Yes, please. Okay.
Yeah,
80, yes, yes, please.
Yeah,
Go ahead.
My question is to Dr. Jazzy, who presented his talk on the chemosensors. So I would be interested to know whether these can be used for, these could be used for even detecting or determining like in scavengers, bacterial scavengers of these heavy metals within the bacterial system. If we can use these chemosensors in some way to detect.
First of all, thank you very much, Professor, for your presentation and for your question. We have not tried this. We have tried in water metrics, and as I mentioned, we have developed a modified version of the protocol in order to detect heavy metal in cosmetics, like in the phonetics, but we have to do it. So I think I will probably we can communicate further if you want to detect in microbial communities and look for the level of metals. So I think this will be a very interesting approach, but we have not thought about this before.
Thank you.
Thank
I will contact you. Thank you.
Great. Thanks. Thank you very much.
So we have no more question, I think.
I think several questions were answered in writing. I think me and my colleagues have answered some of this one.
Yeah. Yes.
Yeah.
I see. Okay.
Sorry, the audience may ask their question via the chat box or Q&A box only, and the panelists can talk via their microphones. Thank you so much.
There is a question on nanotechnology from, I believe, Mr. Anwar Maqdad. So maybe one of the speakers want to answer this. He's asking about future of nanotechnology. Is it more frightening or harmful to humans than beneficial?
Yes, I was wondering whether Dr. Un may want to address that, as it is one of the delivery mechanisms for some of the new medications that are being developed for various pathologies.
So that's the future of nanotechnology. Well, yeah, that's true that nanotechnology has been used as the form of delivery for cancer drugs, but unfortunately, I have not dabbled in that field yet. So I can't provide an accurate answer, I'm sorry.
Actually, I can add some information here on this part.
Yes.
But I thought we have maybe more specialized. So actually, in my research team, we also do nanocarriers for drugs. But so in a way, it's in the nano size around sometimes 100 and sometimes a little bit less than 100 nanometer size carriers. But we use carriers that are biodegradable. So when they are delivered in the system, they deliver their cargo, but then the carrier themselves get degraded. biodegraded by the body. Sometimes if we use metallic nanoparticles, here would be the issue, because these metallic nanoparticles, there is no really clear mechanism on their clearance from the body, especially if you use heavy metal, like silver or iron and so on. So this will have a little bit of an issue with regard to their toxicity. But in general, the nanotechnology is a technology that is here. It has its benefits, and it has some, some unfavorable side effects, and we need to deal with it like any other technology. So it has its beneficial side and also maybe non-favorable side.
May I add
Thank you very
something?
much. Martin, you had your hand up.
Yeah, I think that the question about the future of technology and potential, harmful outcomes
For sure.
is a very interesting one. Sorry?
For sure.
Is a very interesting one in the context of the general pursuit of science. In particular, if you think about our responsibility in research and innovation. And something that some people have suggested is to really think about whether any kind of research projects should be kind of paired or supported by another research project that looks at what the impact of new scientific findings on society are. We see in particular, once some new technology comes out and it needs some regulatory framework, typically the people who build the regulatory framework are typically very late on their job. So is there something that we can do better on that aspect? It's just a question.
Thank you. Professor Azazi, I think you have another question, please, from Zahra Ali.
Yeah, thanks, Dr. Zara, for your question. My sensor apply for detection of mercury ion in water. And as I mentioned, we have developed a modified procedure for detection of mercury in cosmetics, especially the skin whitening cream. And we have developed other sensors by changing the probe itself. Like we use thioketone derivative for detection of mercury. We have other probes for detection of other heavy metals. We have not tried in other matrices. So I think it will be a matter of a protocol in order. So we will have to develop a modified pre-treatment of the sample or the matrix in order to be able to detect mercury in whatever sample we're talking about. But right now, as of now, only water, maybe wastewater, and even for this, we have a protocol. We've tried for cosmetics, but this is all what we have done so far. Thank you very much.
I think Prof. Azazi's presentation has stimulated a lot of interest in terms of other potential applications. I think there certainly is a lot of concern about mercury levels in things we consume and use topically or not. And I wondered whether you wanted to add any sort of general comments, maybe just about safety, or how concerned should we be about mercury in the environment, in our foods, etc.
Thank you very much, Professor Quraisha, for your comment and for your question. I believe also there is a question by Sumaira Sahareen, Dr. Sumaira Sahareen, and she's asking about the use of the chemosensors in plant material like vegetables in order to detect their contents of a metal. Actually, this is a very important question also. We have not tried this again, but the issue here will be the sensor has been developed. it can detect the heavy metal. The issue would be to treat the specimen. Either is it a plant, and then we have to have some sort of a pre-treatment and extraction procedure, and then take the sensor for detection. So this will be the work that need to be modified to handle the specimen or the material that we need to detect mercury in. So I think this should be very straightforward. I don't think this would be complicated. But the sensor itself is very sensitive, very selective, and also has a high absorption capacity for mercury. So I think it should work. But we need to work a little bit on the front part of the procedure to detect, to just extract the mercury or prepare a sample that is suitable for detection for the optimal condition of the sensor. With regard to the comment of Professor Croatia, yes, the presence of toxic metal in our environment is really an issue of concern. And as I mentioned, 4 toxic metal, arsenic, cadmium, lead, and mercury, are on the list of the World Health Organization top 10 chemicals of most concern. So these are probably the most dangerous. So we really need to be more vigilant with regard to release of these toxic metal in the environment, their use in different industries, and the release of the industry. But not only that, as I mentioned, because some contamination of groundwater and well water is due to geological contamination. So the earth itself, or that part of the land, would be rich in these minerals, and this may also contaminate groundwater and well water, and this will primarily affect remote communities. Which they dig a well to reach for water, and they just get this water, and maybe need to check whether or not this water contains toxic metal. And this actually in the US, when some communities there also rely on well water, but they take sample and they send it to the central lab before they use it. And we need to be aware that there are cutoffs for the toxic metal in water that are stated by either the WHO or the Environmental Protection Agency in the US. So there is a certain level for a specific toxic metal at which you should not use the water at all, or another level in which you can use the water for irrigation or for washing, and then another level that you can safely drink the water. So we need to be aware of these regulatory cutoffs, and we need to raise awareness. And of course, before that, we need really to control the release of these toxic metal into our environment, either in air or in water, on our food, and so on. Something also that's very important, which is the presence, unfortunately, of these toxic metal in cosmetics. And this is relevant to women and the Women Conference and Women Day and so on. So some of the cosmetics actually contain very high concentration, not only for mercury, but also of lead and other toxic metal. And I really don't understand why these companies make reasonable profit from selling this. Why do they have to use toxic metal with a high concentration in these products? And of course, this will primarily affect pregnant women, because pregnant women will apply these cosmetics properly daily, multiple times. And this ultimately may reach the baby, and then the baby probably will suffer from lower IQ and so on. So this toxic metal, the most important issue about toxic metal, they bioaccumulate. They enter the body and they accumulate there. And then they damage multiple organs, kidneys, nervous system, blah, blah, blah. So they have wide side effects and toxic effect. So we need to be aware of this. We need to strengthen regulations with regard to use of toxic metal in cosmetics, because some countries, for example, like European Union countries, they have strict regulation for using toxic metal in their cosmetics. Other countries do not. So if they are not regulated, then nobody is looking, and then the danger will be obvious in this case. So again, I think release of toxic metal, we need to be responsible for this. We need to increase awareness about these toxic metal and their side effects, and detection, of course, is very important, because we need to decide whether or not this product contains toxic metal or not, and then we need to avoid if necessary. So I hope this is briefly and just to describe the comment. And thanks very much.
So, okay, Professor Quraisha, shall we end the session?
I think that we've had a great set of presentations. We've answered the questions that have been posed, and I don't see anything new. So I'm quite happy, Prof. Aval, for you to close the session. Thank you.
Thank you very much. So, ladies and gentlemen, may I thank you for your active participation in the session, and I hope you enjoy the program. Have a safe day. Thank you very much.
Thank you very much. Thank you
Thanks,
very much.
everyone. Bye.
Thank you. Thank you, everyone.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
Thank you.